good morning students. Today we are going to discuss about the cytoplasmic inheritance. Cytoplasmic inheritance. It is also known as the maternal inheritance, or it is known as extranuclear inheritance, or the uniparental inheritance. Uniparental inheritance. Okay. The cytoplasmic inheritance or maternal inheritance or extranuclear inheritance or uniparental inheritance. Okay. So we know that if we study the structure of the cell, okay, the cell inside it has the nucleus, and inside the nucleus all the genetic materials they are enclosed. Okay, it is the nucleus inside which all the chromosome or the genetic materials they are enclosed. So this inheritance of this nucleus is known as the nuclear inheritance. But there are certain cell organelles like the mitochondria, okay, the mitochondria or the chloroplast, okay, it is the chloroplast and it is mitochondria. So this mitochondria and the chloroplast, these are the cell organelles. They have their own DNA, okay, and their DNA it is the circular, naked. It is the circular ring like, it is naked, it is primitive type, okay, as like the plasmid. And the second thing, there is a presence of the ribosome, okay, that are 70 years time. 70 years time. So these cell organelles like mitochondria and the chloroplast, they have their own DNA. So these cell organelles, they are present in the cytoplasm and they are inherited to the progeny. Okay, so that is known as the cytoplasmic inheritance. It is also known as the maternal inheritance because at the time of fertilization, when the pollen and the ovule they show the fertilization, okay, so the pollen it has a very little cytoplasm. It has very little cytoplasm, and that cytoplasm they do not have the mitochondria and chloroplast. So only in the ovule there is a presence of mitochondria and chloroplast. That means the large cytoplasm it is only in ovule and it has the mitochondria as well as the chloroplast in them. So it is present in the mother parent only, and therefore it is maternal. Maternal means from the mother. So it is the only maternal. So it is also known as this inheritance only from the mother. Therefore it is known as the maternal inheritance. It is known as the extranuclear because these are the DNAs which are present in the mitochondria and chloroplast. It is outside the nucleus. Okay. So it is extra part than the nucleus. Therefore it is extranuclear, or it is uniparental because it is only from the mother, not from the father or not from the male, only from the female. So it is uniparental inheritance. Okay. So there is the history about the origin of this mitochondria and chloroplast. So that history of origin, that history stated that this mitochondria and chloroplast, these are nothing but the endosymbiotic association. So the association of mitochondria and chloroplast with the plant cell, it is nothing but the endosymbiotic association. Okay, the endosymbiotic association means These are the mitochondria and chloroplast. Earlier time, that means in the ancient time, at the time of evolution, they were the prokaryotes. Okay, they were the prokaryotes. So these prokaryotes, they show the association with the plant cell. So they reside or they live inside the plant cell for their shelter and food, and or shelter or food, or for the protection. Okay, so they live inside the cell. So they are, and both the cell and these prokaryotic organisms, that is mitochondria and chloroplast. So they are mutually beneficial to each other. दोघे एकमेकाला beneficial आहेत. So mutually beneficial to each other, and therefore they are known as the endosymbiotic. So they are the prokaryotes. Prokaryotes, why? Because they have their own DNA, which is circular and naked type. Okay. And second thing, they have the 70S type of the ribosomes. Okay. And these are the characters of the prokaryotes. So these mitochondria and chloroplast, they show the characters of the prokaryotes, and they live inside the cell as a symbiont, and therefore they are considered as the endosymbiont. That means this mitochondria and chloroplast, they are not the evolved part of the plants, but these are the endosymbiotic association of the prokaryotes with the plant cell. Okay, so it is the history of the origin of this mitochondria and chloroplast. Okay, then the most important thing, this extranuclear or cytoplasmic inheritance, it is studied by the scientist Carl Coren in 1908. In 1908, the Carl Coren had done his study on the 4 o'clock plant. 4 o'clock plant. He had done his study on 4 o'clock plant. The 4 o'clock plant means it is the Mirabilis Jalapa. It is the Mirabilis Jalapa. So this Mirabilis Jalapa means it is the गुलबक्ष. मराठीमध्ये याला काय म्हणतात? गुलबक्ष. पिंक कलरचे फ्लावर्स असलेलं या ठिकाणी किंवा व्हाईट कलरचे फ्लावर्स असलेलं या ठिकाणी हे काय आहे? प्लांट. तर Mirabilis Jalapa, that is the गुलबक्ष. So it is also known as the 4 o'clock plant. So Carl Coren had done his study on the 4 o'clock plant or Mirabilis Jalapa plant in 1908, and he observed that this Mirabilis Jalapa or this 4 o'clock plant, it has the 3 types of the leaves or branches. 3 types of branches, and on that the 3 types of the leaves. Okay. So which that 3 types of the branches or leaves? So the first, that are the completely green. Okay, the green branches or the branches with the green leaves. The second, that is the pale or the white. That means these branches, they have the white colored leaves. And the third, that is the variegated. That is the variegated. The variegated means they have the leaves with the patches of white color, okay, and the green color. Suppose it is the green patch. Okay, so these are the leaves. These are the leaves which shows the white patches. Okay. The leaves which shows the white patches. That means this green part it color the green plastids and this white part it contains the leucoplast. Okay. So it is the variegated. So he reported that the green part it contains the chloroplast. Okay, the green part it contains the chloroplast which are responsible for the photosynthesis. Okay, they are responsible for the photosynthesis, so they have ability to produce their own food. And the white patch it has the leucoplast. Leucoplast, that means the white plastids. Okay, these are the colored plastids that are the green. Okay, green chloroplast, and the white colored that are the leucoplast, that means they are the plastid without color. Okay, and they do not photosynthesize. They do not photosynthesize, and due to that these white branches they receive their food from the either green plant, and if the progeny is produced the white, then it gets die off. Okay, and the variegated they have the both these green chloroplast, this white chloroplast, all these they are in mixture, or the variegated they may have the green white or the mixture of the green and white. Okay, so like this the chloroplast or the leucoplast and the photosynthesis, chloroplast and leucoplast, they are present in the plant or that branches. Okay, so he reported that, so we will draw one table here. Okay, suppose it is male, that means the pollen grain. Okay, pollen. Then it is the female, it has the ovule or egg. Okay, and these are the progeny which are produced by this. Suppose the male it is green in color. Okay, suppose male it is green in color, and the female it is the green, pale, and the variegated. Okay, that means the pollen grains from green plant they collected and they are used to fertilize the green branches, that means the flowers on green branches which have the ovule from the green tissues. Okay, then the pollen from green plant fertilized with the pale, and the pollen from green fertilized with the variegated. Okay, that means the flowers on the green branch, flowers on the pale branch, and flowers on the variegated branch. So the resulting progenies were the green, pale, and the variegated. Variegated, green and pale. Next. Okay, so it is the first case. Okay, the second case, when the pollen grains it is from the pale branch. Okay, it is from the pale branch, and the ovule it is from the green plant or green branch, pale branch, and the variegated branch. Okay, so the resulting progenies they were green, pale, and variegated. And the third case was the variegated pollens or the pollen grains from variegated branch, and here the green, pale, and the variegated ovules from green branch, pale branch, and variegated. So the resultant progeny also the green, pale, and the variegated. That means this table shows that Even though the male, it may be either from green or pale or variegated. Okay. The resulting progenies, they were only according to the female. Okay. That means from which female branch or from which branch the female it comes. Okay. That means if it comes from the green branch, the progeny green. When it comes from the pale, the progeny pale. When it comes from the variegated, it is the variegated. Similarly, even though the male it is from the pale, then also if the female from the green, it produces green progeny. If it is pale, it produces the pale progeny, and the variegated, it produces the variegated. Even in variegated also, if it is green, then it produces the green, pale, it produces the pale, and variegated, it produces the variegated. Okay. So these are the results. So Carl Coren reported that the of of the ovule, it has the more cytoplasm. Okay, it has the more cytoplasm, and it has the abundant mitochondria and chloroplast. Mitochondria and chloroplast. But the study of pollenogenesis, it states that the pollen grains, they have very less cytoplasm. Okay, very less cytoplasm, and they do not have the mitochondria and chloroplast. Okay, they do not have the mitochondria and chloroplast. Okay, so this mitochondria and chloroplast, it transmit only from mother to the progeny. Okay, it does not matter the pollen. It may be either from the green branch, or it may be either from the pale branch, or it may be either from the variegated branch. Okay, so it is the cytoplasmic inheritance, or the maternal inheritance, or extranuclear inheritance, or the uniparental inheritance. Okay, so this inheritance, it is from the cytoplasm, so it is cytoplasmic inheritance. It is other than the nucleus, therefore it is extranuclear. It is only from mother, therefore it is a uniparental, and it is from mother, therefore it is maternal. Okay, maternal inheritance. So it is the study of 4 o'clock plant, or study of 4 o'clock, or the Mirabilis Jalapa. It had done by the Carl Coren in 1908. Okay. Then we will draw one figure here. Okay, it is a circle. Okay. Okay. The innermost circle, it is for the ovule, or it is for the female. Okay, it is for the female. So it is for female. This second one, it is for the male. Okay, it is for male. And this third one, it is for the progeny. Okay, so suppose it is this female, it shows the variegated color. Okay, it shows the pale color, and the third one, it shows or it is from the green branch. Okay, ovules from this green branch. Okay, so there are three different branches from where the ovules is produced. So green, variegated and the pale. Okay, so it is the pale. It is green. It is variegated, and the third one is the pale. Okay, then then the male, it may be either green or pale. Okay, green or pale or variegated, green, pale or variegated. Okay, suppose it is green male. Okay, it is green male and the green male. Okay, so it is the female branch. It has the or the ovules. It is from green branch, from pale branch, and from variegated branch. Okay, and then pollens. It is from all these three. That means the pollens from the pale one, variegated one, and the green one. Similarly, here also from pale one, variegated, and green. Pale, variegated, and green. Now the resulted progeny. Okay. Now here the pale. Okay. So if the ovule is pale, even though the male it may be pale or it may be the variegated or it may be the green. So it produce all the progenies they were pale. All the progenies were pale. Okay. So, because the female is pale, therefore, irrespective to the branch of the pollen, okay, the pollen it may be from green branch or maybe from variegated branch or maybe from the pale branch. But then also, if the ovule from pale branch, then all the progenies were pale. Okay. Now the second case green. Okay. So when the female it has the green or the ovule it is from the green branch, then the pollen it may be from the green branch or from variegated branch or from the pale branch, then also all the progenies were the green. Okay, all the progenies were green. And the third case, so it is variegated. Okay. So the progeny is also variegated. That means it produces all three types of the progeny. Okay. Suppose it's green one, then it is a green one, it is green. Okay. Then variegated, variegated, and variegated. Then P, pale, and the pale. That means when the female it is from the variegated branch and even the domain from any of the branch, that means maybe green or variegated or pale, it results or it produces all the three types of this progeny: variegated, variegated, and variegated. Okay, so it is the cytoplasmic inheritance. Then the petite, the next thing, that is the petite mutant in yeast. The petite mutant in yeast. Okay. So the study of petite mutant in yeast, it is on the mitochondria. This study on the mitochondria, as like the cytoplasmic inheritance in the 4 o'clock plant, it is about the chloroplast. So this petite mutant of yeast, it is on the yeast. Okay, so we know that this discovery of this petite mutant, it is by the B. Ifrusi. It had done by B. Ifrusi. Okay, so he had observed that the yeast plant, okay, or yeast fungi, not plant, yeast fungi is the single cell organism, or it is the single cell fungi. Okay, and it has the alternating diploid and the haploid cycle. It has the alternating diploid and haploid cycle. This yeast, it is also known as the Saccharomyces cerevisiae. Okay, it is known as Saccharomyces cerevisiae. Okay, so this yeast cell or the Saccharomyces cerevisiae, it is a single cell organism or single cell fungi which having the diploid and haploid type of alternating life cycle. And this diploid, it reproduces by the ascospore because it belongs to the ascomycetes. Okay, it belongs to the ascomycetes. It belongs to the ascomycetes. Okay, so it reproduces by the ascospores, and this haploid, it reproduces by the isogametes. Reproduced by the ascogametes. So it is the short life cycle of the, or very brief history of the yeast. Okay, so this petite mutant, it is study on the yeast mitochondria. It had done by the B. Ifrusi. This yeast, they are known as the Saccharomyces cerevisiae, botanical name. So this fungi, they are single cell, they belong to the ascomycetes. They have the alternating life cycle, that is the diploid and the haploid. These diploid cycles or diploid organisms, they reproduce sexually by the ascospores and haploids by the isogametes. Okay, so it was observed that these petite mutants or this yeast, they have the mutation. So these petite mutants, particularly that mutation about the mitochondria. So these petite mutants, they fails to these petite mutants they fails to grow on glucose, grow on sugar like the glucose. Okay, they fails to grow on the glucose. Okay, so particularly it is in the case of presence of the oxygen. Okay, so that means these cultures they were developed in presence of the, particularly in presence of the oxygen, and I observe that these cultures they grow very slowly. Okay, in presence of the oxygen they grow very slowly. Okay, so it is concluded that, it is concluded by the we proceed that these mutants they have, the defective, these mutants they have, petite mutants they have the defective, they have the defective aerobic respiratory mechanism. They have the defective aerobic respiratory mechanism. Okay, so their slow growth of the colonies. That means suppose it is a culture plate. So on that the yeast fungi they show develop. So they show the very slow development of the colonies. So colonies they are very slowly developed. Okay. So their slow growth it indicates that they are the less efficient in fermentation. Okay. They are less efficient in aerobic or in the fermentation. Okay, they are very slow in aerobic fermentation. So these mutants they are called as the grande. These mutants, petite mutants, they are known as the grande. So this grande it shows some characteristic features. So what are that characteristic features? Okay. So that characteristic features are, they are first they are insensitive. They are insensitive to inhibitors of the respiratory or the aerobic pathway. Inhibitors of respiratory pathway. Insensitive inhibitors of the respiratory pathway. That means if the cyanides they apply, okay, they are insensitive. There is no any problem is created on that petite mutants. Okay, that means they are insensitive to the pathway, respiratory pathway. Okay, then the second characteristic feature, there is absence of the A, A3 and B. These are the cytochromes. Okay, there is the absence of A, A3 and B cytochromes which are present in the aerobic respiratory pathway. So these three A, A3 and B, these are the respiratory cytochromes which are present in the respiratory pathway. So these three they are absent. That means they have the deficient in the respiratory aerobic respiratory pathway, and many other some changes they are also observed. Then they have incompletely developed mitochondria. The mitochondria they are incompletely developed. And number 4, there is the lack of stenability. That means these mitochondria, petite mutant mitochondria, they do not take the stain. Okay, so lack of the stability in the petite mitochondria. Okay. So this yeast, you know that the yeast, it is we will revise in 1 minute. This yeast, they are the single cell fungi. Okay, these are the single cell fungi which are known as the yeast. Okay, they have the mitochondria, etc., and other cell organelles in them, eukaryotic. Okay, so belongs to the ascomycetes, and they develop or they reproduce by the ascospore when they are diploid, and when they are haploid, they isogamete. They reproduce by the production of the isogametes. Okay, so if Bruce if you see, he observed that this petite mutant, that means some of the colonies of the yeast, they do not develop very well in the sugar culture, or particularly on the glucose. So he had made the study, and he observed that, so there are the some mitochondria, they are incompletely developed. They do not show they have the lack of stability, and they have the defected aerobic respiratory pathway. So they applied the cyanide, and he observed that they are insensitive to that cyanide. The cyanide is a strong, it is a killer. Okay, it is the inhibitor of the poison. So when it is applied, if there is a aerobic pathway, then it should get killed, but it do not get killed. That means they are insensitive to the inhibitors. म्हणजे या ठिकाणी हे सायनाइड टाकल्यानंतर सुद्धा जर एरोबिक पाथवे असेल, तर या ठिकाणी एरोबिक पाथवे असेल तर त्या ठिकाणी ते किल झाले पाहिजे, पण ते किल झाले नाही. म्हणजे एरोबिक पाथवेसाठी ते काय आहे, इनसेन्सिटिव्ह आहेत. त्यांच्यामध्ये A3 आणि B हे 3 सायटोक्रोम्स नाही आहेत. त्यांची डेव्हलपमेंट इनकम्प्लीट आहे, आणि ते स्टेन घेत नाही. तर या ठिकाणी अशा प्रकारचे हे फीचर्स त्या ठिकाणी पेटाइट म्युटंटमध्ये या ठिकाणी दिसून आलेले आहेत. तर या ठिकाणी मग अशा प्रकारचे हे जे कॅरेक्टर ज्या ईस्ट सेलने दाखवले, ज्यांच्यामधल्या मायटोकॉन्ड्रियामध्ये असा फॉल्ट आहे, तर त्याला त्या ठिकाणी काय म्हटले त्यांनी, पेटाइट म्युटंट, आणि त्याला नाव दिलं ग्रँडे. ओके, सो इट इज द पेटाइट म्युटंट. आय होप यू गॉट द पॉइंट. थँक यू व्हेरी मच.